Look at this single biological cell. As its metabolic engine runs, it constantly pulsates, ejecting dark, hazardous microparticles into the surrounding fluid. This process of removing specific metabolic, nitrogen-based wastes to maintain internal chemical balance is strictly defined as excretion. A common mistake in exam preparation is confusing excretion with egestion. Egestion is the removal of undigested food, or feces, from the digestive tract. Excretion deals exclusively with the metabolic waste produced inside the cells. This creates a critical biological dilemma. The body has to purge these toxic nitrogenous wastes without accidentally flushing out vital water, glucose, and amino acids. If this delicate balancing act fails, the concentration of nitrogenous waste in the blood spikes, leading rapidly to systemic toxicity and total organ failure. To solve this precise chemical challenge, human anatomy relies on a dedicated macroscale apparatus, the Human Excretory System. We are going to map the macroscopic plumbing of this system, zoom down into the micromachinery of the nephron, analyze the three-step process of urine formation, and detail the accessory organs that support it. For the WBCHSE exams, pay close attention to the structural breakdown of the nephron. It is the most heavily tested concept in this module. Human survival relies entirely on this relentless dual-scale filtration network to keep the body balanced between extreme toxicity and total dehydration. The entire pathway begins in the abdominal cavity, where the primary components of the excretory system are housed. The system centers on the kidneys, a pair of dark red, bean-shaped organs positioned against the back wall of the abdomen. These two organs act as the body's primary filtration units. Emerging from the renal pelvis of each kidney are the ureters, which are narrow, muscular tubes that connect downward to the bladder. The ureters do not rely on gravity alone. They use peristalsis, rhythmic muscular contractions, to actively transport newly formed urine downward. The urine arrives at the urinary bladder, a distensible muscular sac that functions as a temporary holding reservoir until it is full. Finally, the urethra serves as the terminal duct. It is responsible for the physical expulsion of urine from the body during a process called micturition. These macroscopic structures operate as biological plumbing, solely purposed to manage and transport the chemical product generated deep inside the kidneys. To understand how filtration actually works, we have to look past the kidney's outer cortex and inner medulla, right down to the microscopic level. What you will see diagrammed here is the nephron. It is the fundamental structural and functional unit of the kidney, and there are over 1 million of them packed into each organ. The first major division is the Malpighian corpuscle. This begins with Bowman's capsule, a double-walled cup-like structure that acts as the initial filtration gateway. Housed inside Bowman's capsule is a dense network of capillaries known as the glomerulus. Notice the distinct structural anomaly where the blood enters and exits. The incoming afferent arteriole has a significantly wider diameter than the narrow exiting efferent arteriole. Extending out from the base of Bowman's capsule is the start of the renal tubule sequence. This first highly coiled section is the proximal convoluted tubule, or PCT. The pathway then drops sharply. This is the loop of Henle, characterized by its descending limb, a deep U-turn, and an ascending limb. After the ascending limb, it coils again. This terminal section is the distal convoluted tubule, or DCT. Finally, the DCT connects outward. It empties its contents into a broader vertical central shaft called the collecting duct. This convoluted looping architecture maximizes the internal surface area and extends the time available for complex chemical sorting. Urine formation is an active three-phase process that turns whole blood into concentrated waste. Look at this fully drawn nephron diagram to see phase 1, ultrafiltration. It takes place strictly within the Malpighian corpuscle as blood enters the wide afferent arteriole. Because the exiting afferent arteriole is much narrower, a bottleneck forms. This generates extreme hydrostatic pressure inside the glomerulus. This pressure physically forces plasma through microscopic filtration slits. The liquid that fills Bowman's capsule is called glomerular filtrate. This raw filtrate is a highly volatile mix. It contains toxic urea, but also vital water, glucose, and salts. To prevent the loss of those useful substances, the body initiates phase 2, selective reabsorption. This emergency reclamation occurs as the fluid travels down the proximal convoluted tubule and the loop of Henle. Here, 100% of the filtered glucose and essential amino acids are aggressively pulled back out of the tubule and into the surrounding capillaries. At the same time, passive and active transport mechanisms reclaim the vast majority of the water. This is what prevents rapid systemic dehydration. The fluid then undergoes phase 3, tubular secretion, which is the final chemical refinement step occurring in the distal convoluted tubule. The blood actively pumps remaining targeted wastes, like excess potassium and hydrogen ions, directly into the tubule. Only after these 3 intense phases of chemical editing is the process complete. The concentrated fluid dropping down the collecting duct is now considered true urine. Urine is the meticulously crafted result of aggressive, energy-intensive chemical editing by the nephron. Stepping back to the macro level, the kidneys carry a massive load, but they cannot manage all varieties of metabolic waste independently. The liver acts as the body's primary biochemical conversion center. Inside the liver, a critical process called the ornithine cycle neutralizes highly toxic ammonia. It converts that ammonia into less toxic urea, which is then sent to the kidneys for safe removal. The liver also plays a secondary role by excreting bile pigments, which are derived from the breakdown of dead red blood cells. The lungs function as the body's continuous gaseous exhaust system. Cellular respiration constantly generates volatile carbon dioxide and water vapor. The lungs are tasked specifically with purging these gases from the bloodstream with every exhalation. Finally, the skin operates as a massive thermoregulatory and secondary excretory organ. Sweat is composed of water, sodium chloride, trace amounts of urea, and lactic acid. Releasing sweat cools the body while simultaneously performing mild excretion. True chemical homeostasis requires this entirely distributed network of organs collaborating seamlessly to process specific wastes and prevent overlapping avenues of toxicity. From the largest organs down to the smallest microscopic tubules, the human excretory system operates as a unified, interconnected network. Urine travels a linear anatomical pathway, originating in the renal pelvis of the kidney, flowing down the ureters into the bladder, and finally exiting through the urethra. The ultimate engine driving this balance is the 3-step sequence of ultrafiltration, selective reabsorption, and tubular secretion. Take a close look at this fully labeled diagram of the complete nephron. For your exams, you need to study and memorize this exact spatial layout and the fluid flow directions. The excretory system is the ultimate biological defense mechanism. It constantly filters out internal chemical chaos, maintaining the strict metabolic order required to keep the human machine alive.